impression that you guys are really struggling with this paper.
Yeah,
Okay.
correct.
What's causing you to struggle, other than the fact that it's last school year exhausting, you're just tired as crap? Which is fine. What else is making you struggle? Sonia?
There's just a lot of like letters and concepts we have never learned about in depth, really. And I feel like when I look at the paper, all the letters are swimming at me, and I don't know what the letters
Okay.
mean.
So in those times, it's good to go back to kind of like your core concepts. One of the things we try to stress to you is to get the core concepts down, and they'll help you through other things. So I had this conversation with a couple of groups, but I want to have it with all of you. So what they're trying to do is they're trying to get at the cellular basis for this resistance in snakes. What is it about the snakes that makes them resistant? And they arrive at the idea that it's something about the sodium channel, the structure of the sodium channel. What do sodium channels do? They let in sodium into what types of cells? Primarily.
Muscles?
They'll let it into muscle cells. What other kind of cells? Even more prominent in this other type of cell?
Nerve.
Nerve cells. Okay, so nerve cells. Nerves get propagated by the influx of sodium. Sodium is outside of the cell. When the sodium channel is open, sodium flows in and it changes the polarity of the membrane. It goes from positive on the outside to negative on the inside to more positive on the inside and more negative on the outside. So the polarity of the cell membrane changes. When that happens, sodium channels adjacent to that sodium channel where that change occurred, those then open. That's why it's a voltage-gated sodium channel. Voltage-gated sodium channel responds to changes in the voltage on the membrane. So it starts in one place and it propagates throughout the whole nerve as adjacent sodium channels then open, and then more adjacent sodium channels open, more open, and more open. And that's how the nerve transmission gets transmitted down the length of the nerve cell. So in the paper that you're going to read on Friday, well, I can't really see. In the paper that you're going to read on Friday, they start to look at the snake and the newt across a broader geographic range. So I included in here a map just to kind of set you geographically where they're looking at in this paper. And they're looking at Warrenton, they're looking at Benton, they're looking at Willow Creek outside of San Francisco, and they're looking at this population in Bear Lake, which is where the snake doesn't live with the newt. And what they find when they look at this is that Bear Lake is not resistant at all. Warrenton is kind of resistant. Benton County is pretty resistant. And Willow Creek is super resistant. So they're ranked from not resistant at all to super resistant, and it's the Willow Creek population that is super resistant. And they show you figure 1. Oh, so there's some stuff in here about nerve impulses. When a nerve goes to start a nerve signal, sodium gates open. As they open, sodium diffuses into the cell. That causes more polarization, which causes more sodium channels to open. And so you get this change in the polarity of the membrane from a negative polarity to a positive polarity. Once that depolarization happens, we call it a depolarization. Once that happens, sodium-potassium pumps start pumping sodium out of the cell and bringing potassium in. But for every sodium that it brings in, for every potassium that it pumps out 2 potassiums and brings in 3 sodiums. So, sorry, no. It pumps out 3 sodiums and brings in 2 potassiums. So it's getting rid of sodium faster than it's bringing in potassium. And what that does is it takes the membrane back to a negative state rather than a positive state. And so this is how nerves get propagated, is through the movement of these ions in and out. Sodium channels come in lots of varieties. This is the variety of sodium channel that they're looking at. It's a sodium channel that has 4 domains: domain 1, domain 2, domain 3, and domain 4. These 4 domains get arranged in like little cylinders. that make a pore that goes through the membrane. It's an actual physical opening from inside the membrane to outside the membrane, and that's the opening that allows sodium channels to go through it. It's capable of opening and closing, so when it's open, sodium channels can go through, and when it's closed, they can't. And so there's a pore here in the middle that sodium channels flow through. And this is the one. Patch clamping is just a way of measuring the characteristics of the membrane at a very narrow section of a membrane. And in this case, they used frog eggs, xenopus eggs, which are big so that they're easy to work with. And what you do is you take a really fine pipette, you suck it down onto the membrane, and then there's a fluid in here that will change polarity as it gets more positive ions or fewer positive ions. That's hooked up to electrodes that measure that electrical charge that is on the membrane, so that as sodium channels move into and out of the cell, it registers those changes in polarity. And so it's just a cellular molecular technique for measuring polarity changes on the membrane. And that's what patch clamping is about. There's a video about patch clamping on the Moodle site that you should go and look at for further explanation. This is what it looks like in real life this is the pipette, and this is actually patch clamping on an actual nerve cell rather than patch clamping on something big like a frog egg. So you can do this on really tiny cells. So this is the first figure in the paper. What is this figure showing? Somebody from the high, oh, there's only one person from the high toppers. Garrett, what is this figure showing? You're on the hot seat, you're the only one left.
Yeah. Well, it has each population.
Okay, good.
And then it shows like the change in the, is it mRNA?
It's not mRNA, it's what else? So the RNA would be A, T, G, and C. What are these letters?
The nucleotides.
Not nucleotides. Keep going.
I'm blanking, we talked about it.
We did talk about it. They're not A, T, C, and G. What do those nucleotides get?
Amino acids.
Amino acids, good, yeah. So it's basically showing you for each of these populations where mutations appeared that result in amino acids being different in each of these things. Somebody else, which amino acids are changed in the populations that are most resistant?
Willow Creek.
So Willow Creek is the most resistant population. Which amino acids are changed? There's a change at residue 1556. There's a change at residue 1561. There's a residue at 1568 and 1569. There's not a change at 1566. What happens at 1561? Is Willow Creek the only population that has a change?
No, they all do.
All of them do, yet all of them are still differences, still have differences in resistance. So what does that mean about that particular amino acid change?
It doesn't affect.
Probably doesn't affect resistance at all. Which ones are likely to affect resistance that make this Willow Creek population super resistant? Well, it could be 1556, it could be 1568, or it could be 1569. Either, all of those would be good candidates. This is a change from isoleucine to leucine. This is a change from aspartic acid to asparagine, and this is a change from glycine to valine. How is your amino acid biochemistry?
What? Not so good.
Oh
What's
yeah,
that?
yours is not any better than mine is, and mine sucks pretty bad. So we'll get to what these amino acids actually are like. So what they want to do is they want to ultimately look at the phenotypic effects of these mutations. So how do they go about doing that? Someone from an athlete. are they going to find the phenotype?
It's a great question.
It is a great question.
It is.
Pass it off to another member of your group.
Adelaide. You're in the group,
She's in
Adelaide.
your group. Katie knows the answer to this, I know for a fact, which is why I didn't call on her. Yeah, she passed it up to you. How are they gonna measure the effects of these mutations that result in amino acid changes? What's the method by which they do this? Start back in the very beginning. They're gonna they found the variation in the SNAKE amino acid sequence, which is caused by variation in the SNAKE DNA sequence, right? So how are they gonna get SNAKE DNA to work with?
Did you take it from the fat, something on the skeleton of the snake?
They took it not from the skeleton, but from the skeletal muscle of the snake.
Yep.
And did they take the DNA from the skeletal muscle, or did they take something else from the skeletal muscle?
RNA, and
They
they
take
make it
messenger RNA from the skeletal
in the
muscle.
DNA.
You're in the sports specimens, right?
Yeah.
Yep.
Give them a point. Sticking with these guys, Lily. So, they start with messenger RNA. Then, how do they get DNA out of that? They don't make it into a protein. They want DNA, not protein. They want the frog egg ultimately to make the DNA, to make the protein.
Process it into an.
So in Central Dogma, you generally take genomic DNA, you use RNA transcriptase to make messenger RNA. That messenger RNA gets altered, gets pieces, chunks taken out of it, and then that processed messenger RNA goes out into the cytoplasm, and that's where protein synthesis occurs. So they went to the cytoplasm of the skeletal muscle, pulled out the messenger RNA that's being produced through transcription. And so how do you get from processed messenger RNA back to DNA?
Reverse
What is reverse transcriptase?
processing.
Continue.
Too much pressure.
Okay, too much pressure. It's basically, it's just like RNA transcriptase that takes the template in DNA and adds on messenger RNA nucleotides to make a long string of RNA. You can also use the template of messenger RNA to add on DNA nucleotides to RNA to create a new strand of DNA. What is that DNA called? cDNA. What's the C in cDNA referring to, do you think?
Copy.
It's not chimera, it's a good guess though.
Copy.
It's complementary DNA. What is it complementary to?
The RNA.
It's complementary to the structure of the RNA. That's what the C is about. Okay. Who said something else? Oh, I heard complementary over there also. Yeah, I think it's those guys. So, why would you take RNA? Because one of the problems with RNA, and this is a problem with RNA viruses, with RNA vaccines, RNA breaks down really quickly. You have to keep it really cold, you have to work with it in a really pure environment, a really clean environment. If there's any sort of contamination around, RNA breaks down really quickly for all kinds of reasons. Why might messenger RNA just naturally break down quickly? So part of it could be that it's only one strand, but I'm thinking more of a cellular function reason.
It's temporary.
What do you mean by temporary saving?
Like it's gonna become an amino acid, so like it needs that other part in order to like become whole.
You're getting there. Somebody else. What do you mean by that? Okay, Okay, so that's their method for getting it. When you need a gene product, you can upregulate that gene. They talked about gene regulation at the beginning in the introduction. You can basically turn genes on and turn genes off, upregulation and downregulation of genes. When you need a gene product, let's say that you eat a sugary sweet, you get this pulse of sugar that goes into your small intestine, and you need to absorb that sugar into your cells quickly. What do you produce in your digestive tract that facilitates that?
Insulin.
Insulin. Insulin is a protein, it's an enzyme, and it facilitates the movement of sugar across the cell membrane. But as soon as that sugar is gone, what do you not need anymore? You don't need insulin anymore. So when you need insulin, your genomic DNA gets upregulated to upregulate the formation of messenger RNA that then codes for insulin. But if messenger RNA sticks around forever, what do you keep doing after you've upregulated that first pulse of insulin? You just keep making it, and making it, and making it, making it, and then you die.
Diabetes.
Because you have too much insulin in your system. So messenger RNA doesn't stick around because generally when you need a gene product, you need that gene product for a short period of time. And so you don't want this messenger RNA sticking around forever. You want it to degrade quickly. And so they could have done a lot of things that are easier than getting messenger RNA. Why did they use messenger RNA to get complementary DNA rather than just going to the genomic DNA of the snake to get the DNA that makes the sodium channel? Well, if you can get access to the cytoplasm, you can even get access to the nucleus. You basically explode the cell when you do it the same way. I think you're getting the right idea. I'm just having trouble understanding the way you're phrasing it.
Since the mRNA is complemented with the DNA, it won't break down.
So the cDNA is stable. That's what you always do when you're working with messenger RNA is you're trying to get cDNA because that then is stable and it doesn't break down, so that's good.
Is it because the RNA or the mRNA is like easily isolated, so then you can?
Oh no, it's hard to isolate. It's
Oh.
very hard to isolate. I've done it before. It's a total pain in the butt to isolate. So why did they use this pain in the butt method to get at messenger RNA? It has to do with this. What happens to messenger RNA after it's initially formed?
It gets processed and spliced down.
Okay. Did you guys hear that? It gets processed. So you have a gene of DNA. It's got thousands and thousands and thousands of nucleotides. When you then transcribe that into the initial messenger RNA molecule, that messenger RNA molecule has not only the things that are useful for making the final protein, but it's also got a bunch of junk in it. And one of the things that your cell does is it goes in and it splices out portions of that messenger RNA through what we call post-transcriptional processing to get to the final messenger RNA. And it's that final messenger RNA that actually makes the amino acid sequence. So by taking this post-transcriptional processed RNA, they have the most direct route to the protein. So when they go and put that DNA into an egg of a frog, that frog egg will produce that protein even if it doesn't know how to do the post-transcriptional processing on that gene. It'll just do it. It'll transcribe the cDNA into messenger RNA, and even if it doesn't go through any post-transcriptional processing, it'll still produce the sodium channel at the end. So it's a way of taking error out of the process because they have this chimeric DNA. A lot of you struggle with what chimeric DNA is. What is chimeric DNA?
Made of multiple species.
It's that DNA that's made up of the DNA from multiple species. I don't know, you said something.
Oh, I'm up here.
Oh, you're up there, sorry. So chimeric DNA is made up of DNA from multiple species. What species did they use?
Humans.
They used human DNA and they used snake DNA.
Well, they put it in a frog.
Domain 1, domain 2, and domain 3 were made up of humans, and domain 4 was the snake DNA. And then they put this in a frog. Egg. I refer to this as Frankensteinian abomination in nature. So if you're taking human DNA, you're melding it with snake DNA, and then you're putting it in a frog egg to get expressed and make the sodium channel, you probably want to remove as many barriers to that process as possible. No guarantee that this frog egg will know what to do with human DNA. No guarantee that the frog egg will know what to do with snake DNA. Certainly no guarantee that the frog egg will know what to do with human and snake DNA. So best to just give it the thing that will result in the final product as purely as you can in the hopes of actually getting that to be expressed. Why did they put it in a frog egg? Because they're big, they're easy to work with, and we know a lot about working with them. The Xenopus, the species of frog called Xenopus, they grow them in labs all over the world, and they are a model organism in biology. We have full sequences of their genetic code, so we know exactly where the genes for sodium channels reside in the Xenopus genome. And so what they did was they put these into frog eggs, and then they subjected these frog eggs to tetrodotoxin and saw how well the sodium channels that are expressed by the frog eggs are at being resistant to tetrodotoxin. And that's what these figures are about. Nathan,
That's
let's
the
say something.
phenotype that Dr. Polinsky asked you about at the beginning of this little portion.
Oh yeah, all this work to get to the phenotype that they're interested in, which is resistance. So what do these figures show, David?
Those figures up there?
Yeah, what do these figures show? Figure 8, just start working me through them.
Shows the concentration of TTX.
On the x-axis, yep.
Yeah, and you have the ratio of unblocked to total current.
So ratio of unblocked to total current. So if you're at a high level, what does that mean in terms of resistance?
Your high resistance. No,
So-
yeah, your high resistance.
Yeah, so if you have relatively few channels blocked, then you have high current being produced, and so that would be a sign of resistance. But this individual, this population rather, has a high number of unblocked channels, but only at low concentrations of tetrodotoxin. So is this purple line a line from a resistant population or a non-resistant population?
Non-resistant.
Non-resistant. And what is the yellow?
It's resistance.
More resistant.
It's more resistant. And why do they have so many lines on that?
Population.
Because there's like 4 populations.
So the purple population is Bear Lake, the yellow population is Warrenton. So they only have 2 populations in this figure, but they have 4 lines. There are 2 purple lines that are virtually right on top of one another, and there are 2 yellow lines. What are the 2 lines for each population about? It's in the caption for those of you reading on your screens.
They were trying to calculate the pooled channel data.
Okay, so that's what it is, but what's the difference between the 2 purple lines? What's the difference between the 2 orange lines or yellow lines?
That's the pool.
Nope, that's not the pooled. The pool, each line is pooled for the whole thing?
Channel type,
Hmm?
each channel type.
What do you mean?
And this is the lines represent equations with the estimated dissociation content for each channel type.
And what are the channel types?
Channel entirely snake sequence, and then the trimmer.
Okay, so one of those lines is for a sodium channel that was built entirely from snake DNA versus another channel that was built from chimeric snake human that's what the two lines are about. Are they different from one another? Statistically speaking,
You mean the purple and the yellow or the?
purple and the yellow. The purple lines from each other and yellow lines from each other. Are the human snake chimeras any different than the snake only? Not really, not much. They're slightly significant here, they're not significant at all here. And then of course, purple is lower resistance than yellow. So this is just in these first two populations where they were looking to see if doing this snake-human chimera thing had any major effect on the expression of the sodium channels. Once they do that, what do they have in B? B is just chimeric DNA. And they use chimeric DNA because they know for a fact that the only part of the DNA that varies in the chimeric DNA is that portion in domain 4, the in portion of the snake sodium channel. And what they find is that Bear Lake is less resistant than Warrenton, Warrenton is less resistant than Benton, and Benton is way less resistant than Willow Creek, because Willow Creek still maintains a lot of unblocked channels at relatively high concentrations of tetrodotoxin. Figure C, what does C talk about? What's happening in C? Lily, in figure C.
There is a changing, we're changing a valine to an isolide.
Isoleucine.
That position was also present in 61 in the domain in S5 through S6.
So they're basically asking, at this point, how much of a difference does it make when you go from isoleucine to valine? And what do they find? Are those two lines different from one another? They're different. Are they super different? No, they're different, but they're not that different. They're not nearly as different as the Warrenton population is from the Willow Creek population. Okay? This is the depolarization curve for control versus Bear Lake when given tetrodotoxin. This is the depolarization curve of the control, which is black, versus Willow Creek under a set concentration of tetrodotoxin. And you can't see this unless you blow it way up, but there's a red line and a black line laying right on top of one another, indicating that the Willow Creek population isn't being affected by tetrodotoxin hardly at all, whereas this one is. I should have left myself more time to go through this. We're not done with this paper yet. We will pick up here on Friday. Do you understand more about what the paper is doing now than you did a half an hour ago?
Yes.
Okay.
Very much.
Read the paper for Friday, but also revisit this paper in preparation for Friday so that we can finish up this paper and also get through that paper in the time that we have allowed.
Just like we did for 1989.
All right. I need peer evaluations from my lab. Okay, bring it back. Just shove them on the block on that. I generally am at my in-laws on the weekends. What time is your rest? On Saturday or Sunday? Okay, send me an email about it. I'll see if I can do it. Yeah, thanks. Thank you, sir. Garrett? Thank you, Amanda.
Have a good day. I don't have the right answers though.
I have
did this today.
I,
You want that to
Oh,
be
that's
your
fine, that
grade?
works, that works,
Okay.
yeah, sure.
Is there, can you email me what my grade is
Yeah,
tonight?
yeah, I
Okay,
can do
so I'm gonna
it.
email you to remind you.
Yeah, I can do it as soon as I get back to
Cool.
town.
Thank you.
It's gonna be minus some things that we've
Yeah.
turned in recently, but yeah, I can give you that.
Okay, thank you.
Keep a minor in there.
Where are you going?
We're going to a, plant policy site on Park University's campus.
Cool, I'll come with. I'll see you there.
Yeah, we're gonna meet there at 8:00.
Cool, I'll meet you there.
Well, that was some heavy lifting today.
Yeah.
Did you feel like you did some heavy lifting today?
Yeah, definitely.
Okay.
They were,
They seem to really struggle with this more so than in past semesters, it
just, there were
seems.
two groups they didn't put together until I was sitting next to them, because they didn't become a group.
Yeah, okay, yep.